Okay. So it looks like we're recording. Okay, so I'm here with Professor Sidney Altman. This is a rare honor and privilege to get to sit down with a Nobel Prize winner, Sidney Altman. So you were until recently the Sterling Professor of Molecular, Cellular, Developmental, and Biology and Chemistry at Yale University. And at one point, you're actually the chair of the department at Yale. And among the
I was also the dean of Yale College.
Oh, the dean of Yale College. Okay, that's right. So and then you've had many honors and awards throughout your career. Not the least of which was the being awarded the Nobel Prize in Chemistry in 1989 with Professor Thomas R. Ketch.
No.
Check. Apologies, Professor Check.
That's all right.
Yeah, and you also were awarded in 2016. You were awarded the Lomonosov Medal.
Right.
Is that correct? From Russia. So that's apparently one of the highest honors.
The Russian Academy of Sciences.
It's one of the highest honor they award to any scientist. Like, to non-native Russians or to anyone? Period.
Actually, they every 2 years or so they give the prize out to a Russian and a foreigner.
Oh, okay, I see.
I think that's fairly recent.
I see. Okay, what a great honor, and so you've been kind enough to sit down with us today, and so we can pick your brain and learn a bit about your research and in some way you're sort of like a witness to scientific history. I was looking over your background, and I see that you had opportunity to work with many great scientists in the course of your career, including Francis Crick, Sidney Brenner. Matthew Meselson, am I saying his name? Meselson, yeah. So these were all, to put in context for audience, these were all Nobel Prize winners themselves, right? And so, could you tell us a little bit about your background, like your childhood background? Okay. How did you become interested in science?
I was born and grew up in Montreal. I'm a naturalized American citizen as well as a Canadian citizen at the moment. Through a series of events which were unusual in Montreal, I wound up going to MIT to study physics. I went to Columbia briefly after that, but I didn't like it, so I quit.
Yeah, I read about that. So you were initially a graduate student in physics, is that right?
That's right.
And then what did you not? Why did you quit that? What did you not like about it?
I didn't like it because, first of all, I didn't think I was as qualified as I should be.
Really?
To be a really good physicist.
Well, that's humbling, hearing that, because you were a Nobel Prize winner. So
well, it doesn't matter. The point is that it takes all kinds of talents to.
I see. So you felt you were worried that you weren't right, cut out for physics. I see.
Well, I had trouble handling a lot of the sophisticated equations that one had to deal with. So I quit and went home, and then finally I got a job through somebody I knew at Columbia at Colorado. And while I was there, I met George Gamow. I don't know if you know his name.
Oh, sure, I do. Yes, another famous thinker. Yes, definitely.
That's right. He was Russian. I think he was a great physicist. He was one of the first people to postulate the Big Bang Theory.
That's right.
And the microwave radiation that occurred afterwards. He also explained alpha particle decay. Anyway, I met him at a party for students and faculty at Colorado. And he did something with DNA that I wasn't really familiar with. So we talked about DNA for a little while. And he suggested that if I was interested, I should speak to the people in a small department at the med school in Denver there. Excuse me, I got to blow my nose.
Oh, sure, no, go ahead.
Okay, so I did that, and I wound up, after an hour or so conversation with my person who became my thesis advisor, Leonard Lerman. He was a graduate student of Linus Pauling's.
Oh, I see.
And he was a very, very bright scientist. I did my PhD there.
So you could say that, like, Linus Pauling, you could say is your scientific grandfather. You can make a case for that.
Well, I had no contact with Linus Pauling. Actually, I did have some contact with Linus Pauling many years later. We'll talk about that.
Huh.
So I did my thesis work there. Then I Went to Harvard to work with Matt Messelson as a postdoc, and I spent a couple of years there. And I had originally applied to work with the group led by Sidney Brenner and Francis Crick at the MRC lab in Cambridge, UK, but Sidney had replied to me that they had no room in the lab.
So,
I thought that was a little suspicious. You can always make room for one more person, but anyway, I was in Matt's lab. Sidney came through for a visit a year and a half or so later, and he said, "Do you still want to come to my lab?" I said, "Of course I do." It was the best lab in the world at the time. And he said, "Okay, you can come. We have room now, but you have to bring your own money."
So you have a grant or something?
I applied for various fellowships, and I won one for 1 year to go to the MRC lab. And in fact, Sidney was absolutely truthful in terms of the room. I had as much lab space as the diameter of this table. Okay, to work at.
That's why I mean it's shocking, kind of with the benefit of hindsight. There's like a lot of the discoveries coming out of Messelson's lab. Your discoveries, these are like really like basic, very important discoveries. And there's like, if we could go back, we say should give this guy some more lab space. Like maybe we didn't know what was in the future, perhaps.
No, I think in a reverse sense, the people working under such conditions were kind of proud of it.
I see. So you were forced
to did so much in a very small amount of space.
I see. So you were kind of forced to be creative, and then.
Well, that's a different issue. I had proposed a project to work on when I went to the MRC, and I met with Brenner and Crick shortly after I arrived, and they said, "You can't do this anymore because the problem's been solved. This is the structure of transfer RNA." Which had been solved, let's say, within the past 6 months or so when I got there. So they said, "Go home and think of a new project," and I thought that was stunning. I had to think of a new project which would be approved or not approved by Crick and Brenner. I just didn't know what to do.
Wow, and so were you the equivalent of like a postdoc at that point?
I am a postdoc, yes. And I came up with a project, and I went to speak to them about it. And they were very bored because they thought they had already solved that particular problem, which is the nature of mutagenesis on tRNA. But it depends what you do. They use a particular mutagen, nitrosous guanidine, and I was going to use an acridine derivative. Do you know what an acridine is?
I have to admit I don't.
Okay. It's 3 benzene rings together, and the central ring has a nitrogen in one of the positions instead of a carbon.
I see. And so is this like a teratogen? This is a type of mutagen, or?
It certainly was a mutagen, and it could have been a teratogen. Nobody knows about that. So I worked on that for a while privately, and then that led to the discovery of precursor tRNAs. And once we had a precursor tRNA, it was very easy to isolate an enzyme that cut off the extra piece, so that you're left with a mature tRNA sequence. And that led me into my work on RNase P.
Right. This is the work that you were ultimately awarded the Nobel Prize for. Am I correct? And so actually,
well, I don't know about that. What the prize was awarded for the finding that RNase P has 2 subunits, one of which was RNA.
Ah, I see.
Okay.
So why was that important? Why was this?
Well, I'll tell you, because we found out the RNA was the enzyme.
So there was of the 2 components, RNA was the key component, the one that had
RNA was the enzyme, the protein was a cofactor.
So, but at this point, so not a lot was known about RNA at this point, and so was it just generally believed that only proteins could have enzymatic activity?
Since Pasteur's time, it was thought that only proteins had enzymatic activity.
So you're finding then you cut off the protein portion of RNase P, leaving only the RNA, and that could still get the job done.
No. We didn't cut it off. They're 2 separate subunits.
I see.
They just come together in solution. And we found a way of separating the subunits and testing each one separately for enzyme activity.
I see. And then so ultimately the one that was enzymatic
was the RNA.
RNA, I see. So this was the first discovery of any non-protein acting as an enzyme.
That's right.
I see. Well, that was very exciting. I mean, for one thing, it's fundamental in terms of our understanding of some of the basics of, how, translation can work and things. But also, it raises the question, which I love to talk about, is the ideas of, the kind of the origins of life. Questions about the origins of life.
Yes.
And so, like we think about a cell. Imagine like the first life is some kind of cell kind of crawling out in the egg, but a cell is actually a pretty complex thing.
That's right.
So, I mean, as I understand, there's the theory, the RNA world theory posits that before there were cells, there must have been these kind of, there must have been some kind of self-replicating free-floating units.
Well, I think 2 or 3 years after we showed the enzymatic activity in the RNA, Wally Gilbert wrote a small piece in Nature in which he postulated the RNA world because he said if RNA can carry out enzymatic reactions, and in fact you can show in a test tube anyway that you can select for virtually any enzymatic reaction that RNA would govern in some ways. So he said in the beginning of life you don't need protein right now. You have RNA and you have DNA, and RNA can carry out enzymatic reactions. So you can have some kind of primitive, whatever you want to call it,
Right.
Just made of RNA.
That might have been.
You don't need DNA either, because the RNA can code for information. It can carry out enzymatic activity.
So the RNA is sort of like a Swiss Army knife molecule.
Maybe.
So as I understand, so the RNA maybe perhaps lacks the stability of DNA, but it gains the sort of extra functionality in that it can kind of and you have to forgive me because I'm a behavioral biologist primarily, and so but as I understand, the RNA kind of can loop up on itself and form many different structures.
We're talking about RNA right now and doing different enzymatic activities. Separate RNA molecules can do every one of these activities.
They can do all the normal enzymatic activities that we normally ascribe to proteins.
Well,
or many of them.
Several of them.
Several of them.
That we feel are essential for living organisms.
So why might organisms have why wouldn't they just stick with RNA as enzymes? Like why might organisms have? Why would they evolve to a protein-based enzyme system?
That's a question which nobody really knows the answer to at the moment.
Oh, I see.
You presumably had fragments of proteins floating around in solution at that time. And the question is, how did they get organized so that they could replicate themselves?
Right.
But DNA would be the source of replication. In fact, why did DNA arrive at any particular time? So the thought is that once you could code for proteins, which could be a separate kind of undertaking from RNA, some of the proteins turn out to be more efficient in terms of enzymatic properties than RNA.
So perhaps some kind of natural selection took place, and because they were more efficient.
But you have to. I think a natural selection is important, but we have to understand exactly what natural selection meant. And it's not clear to me that I understand everything about natural selection.
Well, I'm not sure I do either, but I mean, on some level, it's about survival, or being
survival of these very primitive systems.
It's fascinating. Do you know the status right now? Like, what are the thoughts of prominent scientists like yourself and others about the RNA world hypothesis? Like, where is it at right now?
I certainly will not speak about other scientists. A lot of people have different ideas and they have their own justifications for it.
So there's still a fair amount of debate and intellectual
I think there's a reasonable amount of cohesion about the idea that there was an RNA world. Maybe.
So I've heard some people propose that proteins could have possibly been the precursor because we have these strange things like self-replicating proteins, prion diseases, like mad cow disease, or
they're not really self-replicating proteins.
No, why do you say that?
Because they have to be made somehow inside cells.
I see. So they're taking advantage of all the.
they might replicate, they might reproduce themselves in some ways inside cells, but they have to be coded for by some DNA or RNA somewhere.
I see. So they're hijacking an already existing system. They couldn't live independently. There's no evidence they could exist independently.
That's right. That's true of most viruses.
Whereas RNA, there may be can exist independently. Its enzymatic activity is independent of a larger.
No, I think. Excuse me.
Go ahead.
I think that. I don't think RNA can live independently anymore. The only things that you can talk about as living sort of independently are viruses that are coded for by DNA, and there are also viruses coded for by RNA. But in fact, they can't replicate themselves. They have to do it inside cells.
So in a way, we're all, the systems that we have now, they're the survivors of this long process. Maybe natural selection is the right word for it, of millennia, billions of years, and so there are we may not have any like extant examples of this kind of free replicating RNA or independent RNA molecules.
Well, it depends what you mean and how you define what you're talking about. I mean, the fact is that RNase P exists today with the RNA subunit. That's a vestige of what we think was the RNA world. I mean, you can argue about it, but I think that most people will agree that the RNA subunit was there for a very, very long time, and at some time the protein cofactor associated with it.
So when you were awarded the Nobel Prize, was part of the discussion, there's awareness of the potential importance of RNA in the history of life? Was that already in the part? I mean, I know you said that the ideas have been published, about it, but was that a part of the discourse at the time and part of the reason for your Professor Czech's award?
That was talked about as a consequence of these awards. Czech's award was completely different from mine.
I see. Okay.
But these things were talked about as the relevance to the origin of life.
I see. And then, so you were aware pretty early on of the possible.
I was what?
You were aware pretty early on after your discovery that it may have been relevant to early life.
Maybe a year or two after my discovery, because when we were working on this enzyme and what the subunits were doing, we were totally focused on that. I was not interested. It's not that I wasn't interested. I wasn't paying much attention.
I see. So you
were philosophical.
You were very focused on just the proximate details of this challenging system.
That's right.
I see. What were the biggest technical challenges in the course of that discovery?
When I got my first job at Yale, I had in my first year 3 graduate students, and we had already identified RNase P as an enzyme. We didn't know anything about it, period. And I told him to purify it, find out more about it. So that was his thesis project. And after he realized and I realized that we were dealing not with a protein but something that contained probably a piece of RNA subunit with it, that was a challenge in terms of purifying it to homogeneity.
I see.
So his name is Benjamin Stark, and I think he did a very superior thesis once he got over the initial disappointment.
Oh, I see.
It wasn't just the protein.
Actually, I'm glad you mentioned that. In other words, it seems so often when I talk to practicing scientists, like disappointment is like a frequent theme. But what I find interesting is how often, like the great scientists I've met at all scales seem to make lemonade out of lemons. And there may be some initial disappointment, but somehow that pushes them.
The disappointments always teach you something.
Yeah.
It's not as if it's a black hole and everything vanishes. You always learn something from experiments that don't work exactly the way you think they do. And then later on, in 1983, we showed the RNA was an enzyme, but Czech had, in his system, has showed that the year before. But his system in vivo does not have an enzymatic activity.
I see. Interesting. It's fascinating. So you kind of actually, when you mentioned the fact that both of you made a somewhat similar discovery at a similar time, it really brings to mind the fact that so often in history of science we seem to see similar discoveries being made by independent investigators. For example, you mentioned Linus Pauling. Linus Pauling for our viewers is one of the all-time great scientists who was, I believe, awarded Nobel Prize in physics and chemistry, or maybe just chemistry. I can't recall.
What was that?
Did was he a double award winner in physics?
Yes, but in chemistry and peace.
Oh, chemistry and peace. That's right. He became an anti-nuclear war advocate. That's right. But he was just like breathing down the neck of Watson and Crick, as I recall.
That's right. But he had the wrong idea about the model for DNA.
I see. What was his idea?
His idea did not have the double-stranded helix rotating infinitely for DNA. It was a little different.
It was a little different, I see. But he was close. Given him enough time, he probably could have figured it out.
Well, you could say that over maybe to 25 or 30 scientists. Given enough time, they would have figured it out.
Yeah, that's it. I mean, that's interesting. And so you said you actually had a chance to know Linus Pauling.
No, what happened
later in your
was
career,
I was fortunate enough to be elected to the National Academy of Sciences. This was after I had received a Nobel Prize, which I find an interesting little anecdote, to put it bluntly. And a few years later, you get asked to nominate people for various things, and I got asked to nominate people for the Nobel Prize. And I thought that what Matt Messelson has done as a graduate student in Pauling's lab was deserving of that, and the work that he did subsequently. So I wrote a note to Pauling asking him, I didn't ask him to nominate Messelson. I asked him something about Messelson's career. He wrote an angry note back saying, I don't take any advice from anybody.
Wow.
All right. And he died a year or two later. He was in his 90s at the time.
He lived a very long life.
Yes. But his really important work was in the late 30s and 40s.
Right. But ultimately, so Messelson was awarded the Nobel Prize.
No,
he was not. Really? That seems like a terrible injustice, actually.
Well, it's very hard to understand how Nobel Prizes are awarded. But the deal is that the committees that look at people's records want to see something original that contributed to the knowledge in the particular field. What Messelson did was confirm the replication of DNA.
Sure.
And he did it with Frank Stahl.
I mean, it's an immortal experiment that we teach every biology student about that experiment.
I know that. And I think it's important to teach it. But somehow the committee's decided that what Messelson... You're going to have to cut this.
Yeah, no, sure. We can edit it to best effect. Yeah, no need to apologize.
That Messelson had simply confirmed somebody else's fundamental contribution.
I see. It's a shame. It's really interesting. I have no hope of ever winning the Nobel Prize. I'm only hoping to win what's called the Ig Nobel. Have you heard of the Ig Nobel?
Yes, I have heard.
Yes, I'm just hoping. I'm hoping, but I might be the only person in the world who's actually did an experiment in the hope of winning the Ig Nobel. But so far, I'm not even successful in that. I was trying to see if cockroaches are rational decision makers.
Rational?
Yeah, and but mostly,
yes, I think they are.
At most, I barely get a chuckle, and I've given up hopes of the Ig Nobel.
Well, wherever the food is, that's where they'll go.
Yeah, exactly. They are rational. No, yeah, it's a long story. I don't want to distract us too much, but yeah, it is interesting, and I suppose there's a certain amount of politics involved, and there's a lot of, it seems like a lot of hindsight sometimes and debate about the Nobel Prize. May I ask a question about the current science? What current research going on right now, aside from your own research, do you find most interesting, most exciting, in the future of kind of biological sciences?
I'm virtually doing no research at the moment. I'm retired from teaching at Yale, and I'm teaching a graduate course here. if people ask me about the next 10 years or so in biology, I would say that anything that studies how the brain works has to be very, very important. And we're sort of at the edges of learning a little bit about that in neuroscience, but a lot of work has to be done before we understand how thoughts are made and used in various ways.
Yeah.
So I would think that's a very important thing at the moment.
What do you make of there's so there's a bit of a this great discovery of CRISPR technology that's been in
I
the
just
press quite
gave
a
a
bit.
lecture on that.
Yeah, and it's very exciting, and of course I just saw that the scientist who did this horrible experiment to manipulate these children's genomes, that he was fired from the university. It just seems like a very like unnecessary experiment and unethical. But so I mean, in a way, there's two different groups that have a claim to the CRISPR discovery, and I wonder how that will play out.
There's Jennifer Doudna at the University of California at Berkeley, and there's another group in Europe. Now, they did not do the fundamental research on CRISPR.
Oh, no?
No. Most of it was done by people at Georgia.
Really?
And the University of Connecticut, and that's a paper I discussed yesterday or Tuesday with graduate students. You look at that paper, everything is laid out clearly about how CRISPR works. You then can adapt that to mammalian systems and things like that. And that's what Jennifer did, and that's what the people in Europe did. And then they used them to, they obviously didn't alter any human genomes.
No, obviously, yeah.
But there's fighting over who gets priority for the commercial use of a method.
Right, it's a big lawsuit. And so neither of the groups, those two prominent groups, may ultimately be the ones awarded the prize. If you're saying that maybe some other scientists may deserve credit earlier, at an earlier stage of development.
Well, I think if I were awarding a prize, I would certainly give part of it to the people in Georgia.
I see. The University of Georgia.
Yeah.
Oh, I see. Fascinating. And I have to admit, I'm no molecular biologist. So one thing, just to kind of place this in time, like by the time you started graduate school, the structure of DNA had been discovered by Watson and Crick. And so, but is it fair to say that you would have been among the first generation of students in what we now call molecular biology? Is that fair to say?
I think that's probably true.
So that must have been very exciting. I mean, for me, looking back, and like I said, I'm not a molecular biologist, but to look back, I mean, the discoveries in molecular biology are among the most exciting, the most fundamental in any science in the last half century. So there's you had a chance to rub elbows with a lot of these famous figures, and
as you said, I did work with a number of supposedly famous figures during my career, but it was purely a matter of chance. I just met these people knowing other people. I did not pursue them until I had my PhD in a Wangerberg particular lab.
I see.
But it is true that most of the senior people I met at that time originally had been physicists or chemists, people like that. And I had no problem interacting with them and essentially talking their own language with them.
So the fact that you had a background in physics actually was very helpful then.
Well, it was helpful for me in the sense that I had a very rigorous view of data, what to do with data, how to look at it, how to interpret it, how to go on from there. And that came from physics, where you paid attention to everything you did and what, as I said before, what did work and what didn't work.
Fascinating. So there's that gave you an inroad to understand. There's the first generation of molecular biologists who were coming from physics, coming from chemistry. So you had maybe some insight in terms of getting to know them.
I don't know. I'm just telling you about my own experience.
Sure. Yeah. Fair enough. But no, it's really interesting to hear the stories. The kind of the perspective. What was Francis Crick like? What was he like as a person?
Well, the section I worked in at the MRC, as I said, was led by Crick and Brenner, and they shared an office.
They shared an office, like you mean like a lab or like?
Well, they shared a regular office.
Like a regular office.
And which is unheard of in the States.
So they're in a regular office together, like they could like throw
Yes.
paper balls at each other's heads or airplanes.
That's
like,
right.
Hey, wake up, Francis! You're like,
I did not have that much personal contact with Crick,
I see.
but the little I did have and what I heard from other people indicated that he was a remarkably creative person.
I see.
And his early papers showed that. He made suggestions that certainly opened up the field of molecular biology. This was after the DNA model.
So fascinating. And then Brenner. So what's he like? He's still alive, right? He's
93.
93, yeah.
And last week, I believe.
And so wow, 93 years old. There must be something in the water for these great biologists. Linus Pauling, Brenner.
No, Brenner's an absolutely amazing person. He, if you speak to him, he has a very clear line of thought. He knows exactly where he's going and what he's going to say. He has a tremendous sense of humor. And if he doesn't like you, he will use his sense of humor in a sarcastic way, which will cut you down at the knees. Okay?
Sounds disconcerting.
He grew up in South Africa.
Yeah, I think I recall. I remember reading about
And then
that.
he managed to get a fellowship to do a doctoral work at Oxford. And while he was at Oxford, the DNA structure came out. And he and a couple of other guys from Oxford drove down to Cambridge to look at the structure, and he met Crick. And I believe he met Crick at some other point. And
They must have hit it off
that's what I was going to say.
Yeah.
They did hit it off.
to share a lab, or not in a lab, but an office.
Yeah, Crick was mainly a theoretical person.
Really?
But there was one paper published in, I think, 1961 on a genetic code. I think Crick was one of the authors, and apparently he actually did some work in terms of plating out phage and looking at what was going on. There's no question that they got along supremely well, and they produced a tremendous amount of interesting and innovative science.
It's amazing. So it's like a buddy story in a way. The two of them, fascinating.
But then I think it was about 1972, 73, Crick moved to La Jolla.
I see. But I imagine they still were close friends. But it was harder for them to collaborate.
But Crick died about 10 years ago.
I think so. Crick had a lot of broad interests, as a scientist.
Well,
and he was also, I think, interested in neuroscience at the end of his career.
Well, when he left the MRC, essentially devoted himself to neuroscience and to how the brain works. But it was purely theoretical study.
I see. So not necessarily empirical. Fascinating. And so, do you have any words to the wise for young scientists, or for me? I'm actually no spring chicken, but I'm still a young scientist.
No. Whatever words to the wise I have would apply to you and any other scientist I met, except my own colleagues, who I would never implicate in any way. I think that if you're majoring in science at the university, that must mean that you're really interested in some aspect of science. So I would say that you have to work very hard to move ahead. You have to devote yourself to the lab and everything else, and forget about everything else until you graduate.
Just be dedicated to your studies and your science.
And ultimately, try and find a good person to work with.
That's good advice. That is good advice. I looked out that way. Professor Altman, I just want to thank you so much for meeting with me here. Thank you so much. It's a real honor. And so I look forward to. I might have to attend one of your seminars with the grad students and the undergrads to learn about the RNA world and RNA in general. But anyway, thank you so much. It's such a pleasure and an honor. Thank you so much.
Thank you.
Thank you.